Well, good morning, and thank you for joining us. My name is Jim Karrels. I'm the CFO of MacroGenics. I'm very happy to be here with my colleague Eric Risser, our Chief Business Officer. And before we walk you through MacroGenics' pipeline and technology platforms, let me quickly remind you that we will be making forward-looking statements, and we therefore encourage you to review our SEC filings to better understand the risks associated with investment in MacroGenics. So today, MacroGenics is an antibody therapeutics-focused company in the oncology field. We have a clinical pipeline of 9 candidates that are based on 2 of our technologies that were homegrown, including our Fc optimization platform, which enhances antibodies ADCC through some key amino acid substitutions made to the Fc region of an antibody, as well as our DART platform, which is a bispecific technology that we invented to allow us to deploy multiple mechanisms across a wide array of different specificities. In addition, we have multiple program franchises, including PD-1 and B7-H3, in which we have multiple product candidates for each of those underlying targets. We're very well resourced with $233 million cash at year-end. We have multiple alliances across with leading biopharma companies, and we completed the build-out of a 5 by 2,000 liter GMP commercial manufacturing facility last year that we are now actively making drug for ourselves as well as partners. And we sit in Rockville, Maryland. There are 364 employees both across Maryland as well as some folks in Northern California. We have, as you can see, a very robust pipeline of multiple assets spanning different oncology indications with a whole host of targets ranging from novel to very well known. And I'll hand over to my colleague Eric Risser, who will walk you through some of these, and I'll wrap up at the end with some comments on our financials.
Great. Thank you, Jim. I'm going to walk through 5 of our core assets, all of which are in the clinic. And important to note is for 4 of these 5 assets, we've retained major market rights. Each of these also has the potential to be best-in-class molecules, and each of these are in the clinic with major inflection points expected over the next year or so. The first of these assets, margetuximab, this is our anti-HER2 antibody. We're very excited about the recent announcement in February that we had a positive top-line result in our phase 3 SOPHIA trial. I'll describe that study in more detail as we go through some of the slides. We're also gearing up for a broad development effort in gastric cancer, where we're collaborating with a regional partner, Zai Lab, which has greater China rights for this asset to kick that study off later this year. This is employing our homegrown Fc optimization platform. Jim mentioned we've identified some of these specific point mutations in the Fc region, which is the green region of the antibody, that markedly improve immune activation in a broad set of patients. The SOPHIA trial design is summarized on this slide. This is principally a 3rd and 4th line population. All these patients have been pre-treated with Herceptin as well as Perjeta. 90% plus of the patients in this trial also received TDM1 therapy. And the investigators are given the selection of 1 of 4 predefined chemo agents to be the backbone therapy, and then randomized in a 1-to-1 fashion as shown here. We did have a positive result based on the initial readout of PFS data with a hazard ratio report of 0.76, a p-value of 0.033 in the overall population. There are a subset of patients which we define as F carriers that have a low affinity variant for CD16 that typically have much worse prognosis, less efficient engagement of the immune system, where we think with our Fc engineering platform we can further compensate in those patients. And in that F carrier population, we also saw an even more pronounced benefit with the hazard ratio being 0.68 and a p-value of 0.005. We're continuing to accrue the OS events in this study, which have not yet been reported out. We expect that to happen next year, but we are already making plans for a BLA submission, which we are expecting to occur second half of this year. In addition to breast cancer, which in some ways you can think of as almost the worst case scenario setting for an immune-based regimen, late-line therapy in combination with chemo, we're also actively looking at other histologies as well as earlier lines of treatment. Gastric cancer is one where we've already had a very encouraging proof of concept signal in a study that looks at pembrolizumab in combination with margetuximab. So this is a chemo-sparing regimen, which is especially important in this population, which tends to be very frail and debilitated. They do not tolerate chemo, and you'll see the data both in terms of the safety profile but also in terms of the efficacy profile is quite compelling. This study, we had some early data that we presented at ASCO GI earlier this year, which is summarized on this waterfall plot looking at the gastric cancer subpopulation. We did observe a 33% overall response rate in the HER2 positive population, as defined by the 3+ expression. What we also saw in an enriched population of PD-L1 positive, so these are the double positive patients with both HER2 positivity and PD-L1 positivity, we saw a further increase to a 52% overall response rate, and median OS not yet reached in either of these populations. So a very encouraging signal, which if you look at the comparative data of other agents that are either investigational or approved regimens, we'll give you kind of a little bit of lens on that. Starting with maybe on pembrolizumab, which had done their own monotherapy study, yielded a negative study result in the KEYNOTE-61 trial, a 15.8% response rate in a PD-L1 positive subpopulation. Remember, in our PD-L1 positive, we saw more than triple that with a 52% response rate, and significantly improved PFS versus their agent, which showed only a 1.5 month PFS. Importantly, efficacy is an important part of the story, but it's not the only part of the story. From a safety tolerability perspective, frontline therapy is trastuzumab plus cisplatin and 5-FU that is approved with a 70% rate of grade 3 or above toxicities. And again, by comparison, we only have an 18% rate of grade 3 or above toxicities, primarily immune-mediated toxicities tied to pembrolizumab. In second line, which is a comparable line of therapy, ramucirumab is approved in combination with taxane. Many patients, however, opt to avoid the use of a taxane and get ramucirumab monotherapy to improve the tolerability, but you see the ORR drops markedly to only 3%. So we do think this is a really disruptive approach, the ability to have coordinate engagement of both an innate and adaptive immune response by incorporating both an Fc-engineered molecule with pembro to drive very potent activity and very good tolerability. We are looking to move this into a frontline study. We will be also incorporating in that next study our own homegrown PD-1 agents, of which we actually have 3, all in the clinic. One is a traditional PD-1 antibody, and the other 2 are bispecifics that engage PD-1 with another checkpoint, either LAG-3 or CTLA-4. So we'll have more specifics on that trial design as we complete some of the regulatory feedback. We've already had initial discussions with the FDA on the design of that study and are eagerly looking to kick that trial off later this year. I'm going to switch to actually discuss our other Fc-optimized molecule in the portfolio, which is called enoblituzumab. This program, however, is directed against a target called B7-H3, a very novel target where there's been really an explosion of literature over the last couple of years, both highlighting its broad expression patterns across multiple solid tumors. We've also seen that there's a clear immunologic role of this target in terms of driving T-cell immunomodulation. In our recent IST that we've conducted in prostate cancer, looking at a neoadjuvant population, we've also seen that monotherapy treatment with enoblituzumab can in fact drive increase in CD8 positive T-cells. So there's clearly the ability to drive a crosstalk of both innate and adaptive immunity with this molecule, given that it has both the enhanced ADCC component, but also this T-cell immunomodulatory component. This molecule, again, highlighted on this slide, FC optimized. We are going to be kicking off also a development effort in a frontline population looking at head and neck cancer later this year, so that planning effort is well underway. The early data which we presented at SITC, which gave us a further conviction that there's a real development path here, is highlighted on this slide. So this was a design of a combo trial, enoblituzumab plus a PD-1 agent. Here we use pembrolizumab, and what we highlighted is two specific populations which were checkpoint naive, both a head and neck cancer population as well as a lung population. In the lung setting, however, we were further restrictive, and we looked at only patients that were PD-L1 negative, which we know is an even higher bar, and where pembrolizumab and nivo have not had as significant activity profiles. The data in head and neck is summarized here in a waterfall plot where you can see nice activity across both the HPV negative as well as HPV positive subsets. We observed a confirmed response rate incorporating both PRs and CRs of 33%, which was further enriched when we looked at a enrichment in terms of B7-H3 expression in the tumor of 10% of the cells having 2+ or 3+ expression based on immunohistochemistry. You saw a closer to a 40% response rate in that subpopulation. You can also see, as illustrated in the upper insert, the spider plot, where many of these patients either still on therapy and have had, in many cases, quite a long duration of their response. A similar profile was apparent in our lung cohort, where we had 14 evaluable patients, again, an overall response rate of about 36%, further enrichment that led to a 45% response rate in that higher B7-H3 expression patient population, and again, nice durability in these responses as well, and this was both across the squamous and non-squamous histologies. To put this into perspective, we provide some benchmark data on this slide. The upper few rows are highlighting the head and neck experience and comparing it to the CheckMate and Keynote studies conducted by BMS and Merck. Most of those are in the mid-teens range, and again, we're at a 33% response rate. Lung, depending on the histology, 9 to 17%. So here again, we're looking at doubling, if not quadrupling, of the activity profile. Important to note here, not beyond these two experiences, margetuximab is really the third experience where we've had an FC-optimized molecule in a checkpoint-naive population in combination with a PD-1 agent, delivering activity profiles that are double, if not triple, of what PD-1 monotherapy has shown. So we're very encouraged by this, and again, are going to be kicking off a phase 2/3 adaptive trial design initially in the head and neck cancer setting, targeting a frontline patient population. This is also not the end of where we're going with this target. So enoblituzumab is the lead asset, but we have two other molecules which we think have complementary mechanisms. We have a DART molecule that employs the redirected T-cell killing modality called MGD009, which is going through dose escalation in both monotherapy but also in a PD-1 combo study. And we have also our first ADC, which is also now in the clinic going through dose escalation. This is leveraging a proprietary drug linker that we licensed from Synthon Pharmaceuticals. It's the same drug linker that they've actually taken into late clinical studies of their own, so we think there's been already some clinical validation here. And very excited about these programs, and also the fact that they each bring really a different mechanistic basis to the table. Enoblituzumab leveraging the innate immunity component, driving enhanced ADCC, which is primarily mediated by NK cells. 009, which is really focused exclusively on redirection of T cells, and then 018 is more of a direct tumor lysis by internalization of this very potent DNA alkylating agent called duocarmycin. 012, this is a PD-1 agent. It's a hinge-stabilized IgG4 antibody that compares very favorably with some of the approved agents. It's been actually engineered to have even higher binding affinity for PD-1, a slower off rate, better blockade of PD-L1. That's been on the preclinical engineering piece. In the clinic, the expectation is this will perform as good as the approved agents. The early data that Incyte has presented seems to corroborate that across multiple different tumor types. We, as part of this relationship with Incyte, have retained rights for leveraging this agent for combo studies with any of our internal pipeline assets. We also continue to provide global clinical supply for this program for all of our studies as well as all of their studies. And there's a pretty abundant list of trials that are now being conducted. I actually looked at clinicaltrials.gov earlier today. There's now 10 studies listed, one of which just came on board this week. So they are not just pursuing monotherapy, where they have 3 studies with registration intent that are ongoing, targeting MSI-high endometrial, Merkel cell, as well as anal cancer. The data from these studies are expected to already start coming available next year, with hopefully approval not too soon thereafter. On the combination front, which is really one of our core development theses, is to have our own PD-1 agent to enable very broad and easy combinations. So we're doing combos with many of our pipeline assets. They've already initiated trials with both epacadostat and their PI3 kinase. They've also supplemented that with more recently trials targeting FGF and their arginase inhibitor. So there's a proliferation of studies, and again, irrespective of which study wins and ultimately yields an approval, we also garner significant development milestones, sales milestones, and a tiered royalty rate that goes from 15 to 24%. That same specificity in terms of the PD-1 binder that's in the antibody, we've actually also incorporated in our PD-1 LAG-3 bispecific molecule. So this is a single recombinant molecule that provides co-blockade of these 2 checkpoint molecules. This is now recently completed a dose escalation. We're very excited about the profile of this molecule. We're actually taking it forward in 11 separate expansion cohorts that span 9 different tumor types. We do have global rights for this molecule with the exception of Greater China. So the recent deal that we entered that I alluded to with Zai Lab encompasses both margetuximab and the MGD013 asset. One of the exciting features of this molecule, which I think really differentiates it from a lot of the LAG-3 development efforts that many of the big pharmas are embarking on, is the fact that we have that single recombinant molecule with this tetravalent format that engages these 2 molecules. BMS, for instance, they are actually doing a frontline gastric study in a HER2-negative population. They've done quite a lot of development efforts, but it's been with a PD-1, the Nivo molecule, with their LAG-3 specificity. And you can see, based on the benchmarking here, looking at an interferon gamma induction assay as a kind of marker for T-cell activation, that if you start from the bottom, we have a control across a varying set of concentrations. We have the BMS LAG-3 specificity, which as a monotherapy, more modest activity, which is consistent with the clinical experience. Our LAG-3 specificity can recapitulate that T-cell activation. Nivo, as a monotherapy, towards the middle of this chart, shows a improvement, which we can again recapitulate with our O12 assay. It's really the combination of the 2 where you see a more marked improvement, which we can recapitulate by just taking 2 of our antibodies. But on the very top panel, you see a very significant improvement even on that. And the notion here is you have a tetravalent molecule with the avidity advantage that you can co-engage both of these checkpoints. There tends to be also more persistent expression of PD-1 early in the cycle of T-cell exhaustion, whereas LAG-3 in some cases comes up late. So if you can actually anchor in early on with that same molecule, you have a better chance to provide co-blockade. And again, it translates in these preclinical settings with substantial improvement in T-cell function. Flotetuzumab, another molecule, one of our kind of big 5 that will be having a real substantial inflection points this year. This is one of our first DARTs that went into the clinic, leveraging the redirected T-cell killing mechanism. One arm engaging CD123, which is a leukemic stem cell target that does have limited expression patterns on normal hematopoietic stem cells. And then the other arm is CD3 in terms of providing recruitment of T cells. The lead indication here is relapsed refractory AML. We had some data we presented in an oral presentation in 2018. We'll have additional updates later this year at ASH. This molecule is part of a partnership with Servier, where they have rights in Europe and some other geographies, but we've retained full rights in both North America, Japan, as well as Korea and India. So the design of this study is shown on this slide, where we've gone through an expansive dose escalation phase and 2 separate dose expansion phases, where we're really trying to further fine-tune the scheduling of this molecule, how to titrate the dosing upward to spare and blunt some of the cytokine release. That's really endemic to all these T-cell redirection modalities, as well as thinking about how to use supportive care, things like steroids and anti-IL-6 therapy in a way to further curb the cytokine release. We think we're making significant progress on that front and expect later this year to have a schedule, a dosing schema, as well as a potential registration path that we can articulate to our investors. The data that we presented at ASH 2018 is shown in this waterfall plot. You can see very good activity, about a 30% rate of CR or CRI in a primary refractory subpopulation. So these are patients that have failed 2 or more induction attempts or had a CR with a relapse after a relatively short response. And this is a subpopulation where the benchmark data suggests about a 13% CR/CRI rate, so significant improvement there. And again, with some of the measures that we've taken more recently to provide a more tailored and gradual dose escalation within each patient, as well as how to leverage anti-cytokine therapy, we think we're going to also have even a more pronounced advantage in terms of management of CRS. So stay tuned, but we'll have additional updates on this program later this year. And with that, I'm going to turn it back to Jim Karrels to walk you through some of the financials and some of the key milestones.
Thanks, Eric. As I mentioned at the outset, MacroGenics is well capitalized to pursue all of these opportunities before us. We ended the year with $233 million in cash. Subsequent to year end, we brought in $22.5 million from our partnership with Zai Lab. In addition, in February, we completed a financing of sale of our shares, raising an additional $118.5 million, bringing our pro forma cash total to about $374 million. As you can see, our operating expenses have been increasing over the years, as you might expect, with a phase 3 study, sort of midstream right now, or on the tail end of that. And we expect that with the addition of other product candidates coming into our pipeline and moving into later stages of development, this trend will likely continue. I will note in the bottom panel that we have been able to bring in a significant amount of non-dilutive capital from our partners. Since our IPO in October of 2013, we've brought in more than $450 million in cash. And of course, all of this sort of pays for the anticipated progress progress we've not only made thus far in 2019, but that we expect to make through the remainder of the year, including the various programs that Eric walked you through. And just to quickly tick through those again, we had a positive top-line margetuximab data in breast. We expect to initiate the gastric cancer study with margetuximab in O12, second half of this year. Enoblituzumab, we will initiate a study with MGO12 and Enoblituzumab in head and neck cancer later this year. MGO12, Incyte is running the program there, but we also obviously have other combination studies ongoing. O13 data should be in the latter part of the year, and flotetuzumab as well, the CD123 by CD3 program. We have additional monotherapy data that we'll present before year end. And with that, I thank you very much for your interest in MacroGenics and your time today. Thank you. I think we have time. Hello, Kime.
How are you?
Great.
Just remind me again sort of about the pathway to market for at this point in the month. What trials do you need? What are you thinking about? Are you doing a combo trial?
I'm sorry, for margetuximab in gastric or in breast? Both.
So breast, obviously, we had a positive endpoint in terms of hitting the PFS. So the expectation is that we will submit our BLA filing second half of this year, and if approved, obviously we'll be in a position to commercialize next year. Gastric cancer, we have had some early interactions with the FDA. Our partner Zai Lab is also having some outreach to the Chinese regulatory authorities to solidify that trial design. That would be initiated later this year. We have not provided guidance in terms of when that data would be reading out in terms of enabling a submission.
And are you, at this point, committed to taking it alone to the market for breast, or partnering, or what's the thinking around that?
We're exploring both. Obviously we see this first readout from SOPHIA as being a starting point. We see there's an opportunity to really have significant expansion in terms of reach, as well as go into earlier line populations where we think an immune-based mechanism could have an even more pronounced benefit. Obviously, being able to support all of that will require additional help with the partners, so that's definitely actively being considered.
And then on the B7-H3, can you just walk through a little bit of the competitive landscape as well as really developing B7-H3?
There are a couple other agents in development. One is from Y-mAbs that has a radio-labeled antibody that they're taking forward in a kind of a pediatric cancer. There are also other ADCs that have been disclosed publicly, actually including Daiichi Sankyo, which has a preclinical program that they're developing. AbbVie has also a ADC directed against B7-H3 where they provide some updates at AACR recently. So there's definitely a target that is having growing interest, just again based on all the literature and work being done on that.
And then over the next year or two, this is one question I have for you, is there a way to offset the spend through partnerships? Is that a goal to try to do much more with earlier stage pipeline development and sort of get more funding through?
Yeah, the question is with regard to, well, first cash runway. So the cash that I mentioned, $374 million pro forma cash, takes us through 19 and 20. It's into 2021. Historically, as you can see on the bottom panel, we've had pretty good success annually in partnering sometimes very early, preclinical assets, sometimes phase 1 assets with big pharma and big biotech. We certainly hope to continue to do that. When I say that our cash takes us into 2021, into 2021, we have, I think, fairly conservative assumptions underlying partnering, de novo new partnering from different companies.
And just last question from me is on the B7-H3 side, aside from data that you can produce, is there data from other companies that you feel is really important to the space in order to understand the value of the class that we should be looking for?
I think at this point we're primarily fixated on our own data with, given that we have 3 molecules, each has a different mechanism. Some of the other competing efforts in this program are more predicated on just a direct killing mechanism, leveraging the fact that you have such a broad expression pattern of this target on many different solid tumors. Obviously, one of our lead programs, enoblituzumab, is more predicated on an immune modulatory effect in addition to that. So, each one has a unique profile. And our near-term focus is to get the phase 2/3 study kicked off later this year.
Thank you. Any other questions? Thank you.